Hello everyone, welcome back to my channel. So with this video, I'm going to start with a new topic, that is forensic odontology. I'll be discussing what forensic odontology is, that is a small introduction will be given. Then who is the father of forensic odontology, a book and a paper written by him that has been asked in a previous year entrance exam. Then the factors for dental identification, what are the various factors that are used for dental identification, and then the role of a forensic odontologist. Also, if you want this PPT, then you can go check out my website. I have provided this PPT over there. The link for website is given in the description box. Now let's get started. First, let's understand what forensic odontology is. So forensic odontology is the study of dental application in legal proceedings. So as the name is suggesting, forensic odontology. Odontology is the study of teeth, right? That is the dental study. And the forensic, we know what forensic is, right? Forensic is the application of various sciences like chemistry, physics, dentistry, medical science, right? All these sciences for legal proceeding or for providing justice, right? So if these two words are clubbed together, forensic odontology, then it is basically the use of now what kind of application of what kind of knowledge, that is dental knowledge or dental study, in order to solve forensic cases, right? So now in forensic odontology, what we do is there's proper handling, examination and evaluation of this evidence, that is the dental evidence, which will be presented in the interest of justice. So basically the dental evidence is properly handled, examined and evaluated in forensic odontology. The evidence that may be derived from the teeth can tell us about the age of the person and also can help us in identification of the person to whom the teeth belong. Then the father of odontology is Dr. Oscar Amino, not A but this is O. Dr. Oscar Amino, it's written here, and this is a picture of Dr. Oscar Amino. He is known as the father of forensic odontology. There's a famous paper written by him, that is, I don't know if I'm pronouncing it right, but the name of the paper is The Bazaar de la Charite, and it was written in the year 1898. And also there's a famous book written by him, and this is also the first book that is written on forensic odontology. I don't know if the pronunciation is right, but Les Arts Dentaires en Médecine Légale. This book was written by Dr. Oscar Amino and it was published in Paris in the year 1898. And also this is the first book on odontology, forensic odontology. Here is a picture of the book also. Now, forensic odontology is proved to be very much beneficial, especially in the cases of mass disasters. So, forensic odontology is of immense help in identification of disease in mass disaster situations like earthquake, tsunami, air crash, etc. and in the identification of decomposed and disfigured bodies like that of drowned person, bomb blast, fire victims and victims of motor vehicle accidents. So forensic odontology talks about what? Teeth, right? And teeth are the hardest portion in our body, so they resist any kind of destruction. So in cases of mass disasters, even when the body of the person is completely destroyed, what we get to see is teeth are still present. Teeth may be still present in intact position, so hence these teeth can help us even in cases of mass disasters like earthquakes, tsunami, air crashes, etc. Also, they decompose at very late stages, so even in severely decomposed bodies, teeth can help, or in disfigured bodies, these teeth can help us identify the person. Now, there are various factors that can be used for dental identification. The most basic factor that we can use is count the number of teeth. So, the assessment of teeth in the mouth should include those that are present and those that are missing. The number of possibilities that exist for the combination of teeth missing or present may well be enough to obtain a positive identification, including those that are present or missing. So if we know how many teeth a person had before he or she died, we can count the number of teeth with the diseased body that we are having, and we can get an idea. Although this method does not give us any conclusive result, but yes, we can use it. Like for example, in few people, we do not get to have the third molar, right? Not everyone gets the third molar, right? So that can help us know if the dead body is having a third molar, whereas the missing person does not have. So we can ascertain that okay, this is not the person that's missing or that we are looking for. Then restorations. A tooth is restored when there is a loss of tooth substance due to dental caries, fractures, or some abnormal wearing of the teeth, or for an aesthetic reason. The type of filling material used. So basically, people restore their teeth for various reasons. Like for example, if there is any kind of fracture in the tooth, or any kind of wear and tear of the tooth, or even for aesthetic reason, just for eye pleasing reasons. So people can go for filling materials, and there are different kinds of filling materials that can also be used. Like the most common one is silver amalgam. Then there can be precious and non-precious metals can be used. Then crowns of gold or porcelain fused to material can also be used. Temporary dressings or fillings may also be used that are made up of zinc oxide, eugenol. So here you can see restored tooth are there. Like for example, here it's gold, here it's silver. So if any person have had restoration of the teeth, then that becomes even more easy to identify that person. Then dental prostheses, like a full denture, removable part, denture, bridges, orthodontic appliances. So dental prostheses or dentures like these, if the person is using, like in old age, right, people use dentures or dental prostheses, then that also helps very much to identify any person. Then the bony pattern. The medullary bone of the jaws possess a characteristic trabecular pattern which can be duplicated on the post-mortem radiograph. Tooth angulation, root morphology, bone loss, specific change in pulp chambers, pulpal outlines may also be assessed. So bony patterns, everyone has a characteristic bony patterns. That is, we can talk in terms of tooth angulation, root morphology, bone loss. All these factors can help. Then the occupation, the habits, and the social position of the person can also help modify the teeth of a person in one way or the another. For example, severe generalized tooth attrition is common among workers who are continuously exposed to atmosphere that contains abrasive. Then there are certain habits that may also leave some kind of permeable vestiges that may help in develop a profile of the individual. Like if someone is used to chronic pipe smoking or long-term backpipe playing, then they might have unilateral bimaxillary wear and tear pattern, and notching on the upper incisors. So if we know the daily habits of those person, or the kind of environment that they are exposed to, there can be attrition seen, or there can be wear and tear due to continuous smoking or playing some kind of instrument like bagpipe. We can compare the teeth and then link if that's the same person, or if that can be same person or not. Then there are various roles of forensic odontology. First is the identification of unknown human remains through their dental records. Then providing assistance to the forensic experts and police personnel in cases of mass disasters. Then age estimation of both living and deceased person, that includes neonatal remains. There are various methods for this age estimation that will be discussed in my upcoming videos. Then there can be analysis of bite marks found on the victims or accused in case of physical or sexual assault. Then identification of bite marks on substances such as wood, leather, foodstuff, etc. Then forensic odontologists can also help in the identification of weapon of offense using the same principle of bite mark analysis. They can also act as an expert witness in cases involving bite or weapon mark. They can also provide assistance to archaeologists in building up a dietary history of tribe or a race. So all these are roles of a forensic odontologist. Four types of teeth, that is incisors, canine, premolar and molar. So this is a picture of all the four type of teeth. This is how an incisor look, this is how a canine look, this is how a premolar look, and this is how a molar look. So in the upcoming slides, I'll be discussing about each one of them, all the important points that you must know about these teeth, right? Also, if you want this PPT, then you can go check out my website. I have uploaded this PPT over my website. The link for website is given in the description box. Now let us study them one by one. First, let's start with incisors. So incisors are 8 in number. So there are a total of 8 incisors, and they are present in front area of the mouth. Here if you look at this picture, this teeth and this teeth and this one and this one, and here this and this, these are the total number of incisors that are present. And there are 4 in the maxillary jaw. Maxillary teeth are the teeth that are present in the upper portion, like in this particular portion, the teeth that are present, they are maxillary teeth, whereas in this lower portion, the teeth that are present are known as mandibular teeth. Let me just write down, this is mandibular, all these are mandibular teeth, whereas the upper ones are the maxillary teeth. So the center one, these two are central incisors. Similarly, these two are also central incisors, whereas the one that are present next to central incisors, that is this one and this one, these are the lateral incisors. Okay, so incisors are of two types, central incisor and lateral incisor. Incisors are shovel shaped. You can see the shape in this picture. They are shovel shaped, like they're shaped like this, like a shovel, right? They have a single root, as you can see here. This is the root, and it is single and it is conical in shape, right? And incisors have sharp incisal edges. For this teeth, this is going to be its edge. In case of incisors, the edges are sharp and hence they are used for cutting purposes. Incisors are the teeth that is used, that you use to bite your food. So this is the role of incisors. And the smallest teeth among all the teeth are mandibular incisors. Mandibular means lower, the incisors that are present in the lower area, that is these incisors, they are the smallest teeth in your entire mouth. Next, let's talk about canines. Canines are the next teeth that develop in your mouth. They are present between incisors and premolars. So till now we know that these are incisors. So canines are present next to incisors. So this one is canine, and here this one is canine. Similarly, this is canine and this is canine. Alright, so we have a total of 4 canines in our mouth. Okay, they are V-shaped. Here look at this picture, this is kind of a V shape. So canines are V-shaped, and they have a cusp. This area is known as the cusp, right? And canines are also known as eye tooth or cuspids. They also have a single root only. They are the sharpest teeth and hence they are used for tearing the food apart. Amongst the canines, upper canines are the longest, that is the maxillary canines are the longest. Now let's talk about premolars. Premolars are used for tearing and crushing of food. Unlike incisors and canines, premolars have a flat biting surface. Look at this picture. These 4 are the premolars. 4 premolars are present in the upper jaw and 4 premolars are present in the lower jaw. So there are a total of 8 premolars in our mouth, right? So there are 8 premolars in total. This picture is showing just the just the mandibular teeth, and similarly they are going to be maxillary teeth also. They are also known as bicuspids. First maxillary premolar has 2 roots. First maxillary premolar, alright, they have 2 roots, whereas all the other molars have only 1 root. So if you look at this picture here, you can see that there are 2 roots. That means this one is a first maxillary premolar. A premolar might have 3 to 4 cusps. Maxillary first premolar has 2 cusps, whereas mandibular first premolar also has 2 cusps, and the mandibular second premolar has 3 cusps. So this is the shape of a premolar, and this is the location of a premolar. Here we have the central incisors. These are the central incisors, these are the lateral incisors, these are the canines, and this is the first premolar, second premolar, first premolar and second premolar. Alright. Then if we talk about the molars, so molars are the largest teeth in our mouth. Their function is similar to that of the premolars only, that is to grind, tear and crush the food. Molars have large flat biting surface which makes them perfect for this job, for the job of grinding, tearing, crushing food. Typically there are 2 roots in molars, but maxillary first molar has 3 roots. Here's a picture of how a molar looks like, and in this particular picture these 3 are the molars. Alright, this is the first molar, this is the second molar, and this is the third molar. As we know, third molar arises the most late, that is it arises in between 17 to 25 years age, and it might not even arise in some people. It is also known as wisdom teeth. The third molar is also known as wisdom teeth. So molar typically has 4 to 5 cusps. The maxillary first molar sometimes have 5 cusps, and it is known as cusp of carabali. This point is important. Generally there are 4 you can say cusps, but sometimes what happens is that the maxillary first molar, that is the upper first molar, sometimes might have 5 cusps. So that cusp is known as it has been given a special name called cusp of carabali. This is an important part. So these were the 4 teeth, that is incisors, canines, premolars and molars. I hope you understood all the different types of teeth. The structure of teeth in humans, as we know that human teeth has many parts like enamel, dentine, pulp, root canal, etc. So all these parts will be discussed in detail in this video. Also, if you want this PPT, then you can go check out my website. I have uploaded this PPT over there. Link for the website is given in the description box. Now let's get started. Firstly, let's understand that our tooth is divided into 3 main parts, that is the crown area, root area and the neck area. So a tooth is divided into crown, which is the part above the gum line. Here you can see crown. This is the area, crown area, right? It is present above the gum line. Then comes the root area, that is the lower portion, and this is the area that is present below the gum line, and in between these 2 area comes the neck area, right? So this is the crown area, which is basically the visible area, the one that we can see from our eyes. Then comes a neck area that is present between the crown and the root, and then is the root area that is present under the gum area. So a tooth comprises of enamel, dentine, cementum, dental pulp, periodontal ligaments, jawbone and gingiva. All these structures will be discussed one by one in the upcoming slides. So let's get started with enamel first. Enamel is the hardest part of our body and it is the most mineralized body part. 90% of this enamel is made up of minerals, with water and organic material comprising of the rest. So 96% is mineral, whereas 4% is water and organic material. If you look in this picture, enamel is this area is the enamel, right? All the white portion is the enamel. The portion that we can see through our eyes is the enamel. So the enamel covers the crown of the tooth. This is the crown area and it is covered by enamel. As you can see, this is the dead portion. It has its origin from ectoderm. So we know that there are different germ layers like ectoderm, endoderm, mesoderm. So enamel comes from the ectoderm germ layer. It is normally the visible and it is supported by the underlying dentine, which is the next part that I'll be discussing. The normal color of the enamel, it varies from light yellow to grayish white. The thickness of enamel, it varies over the surface of the tooth and it is often thickest at the cusp area and thinnest at the border. The wear rate of enamel is known as attrition. Enamel's primary mineral is hydroxyapatite. You must remember this is the main mineral that enamel is comprised of. And hydroxyapatite is basically crystalline calcium phosphate. The largest amount of minerals in enamel accounts not only for its strength but also for its brittleness. Enamel does not contain collagen. So these are all the important points about enamel that you must remember. It is the hardest body part, most mineralized. The mineral that it is made up of is hydroxyapatite, which is basically crystalline calcium phosphate. Enamel origins from the ectoderm layer. The color varies from light yellow to grayish white. Enamel is thickest at the cusp and thinnest at its border. So these are the few points. Now let's talk about dentine. Dentine is a substance that is present between the enamel and the pulp chamber. This area is the dentine. As you can see, it is present between the enamel and the pulp chamber. It is secreted by the odontoblast cells of the dental pulp. The formation of dentine is known as dentinogenesis. We know that genesis means formation. So the formation of dentine is called dentinogenesis. Dentine is a porous yellow-hued material and it is made up of 70% inorganic material, 20% organic material and 10% water by weight. It has collagen. When a cavity forms in the enamel and continues in the dentine, it progresses very rapidly to the pulp since the dentine is very soft, due to which we get to feel sensitivity in our teeth. Dentine is the alive portion. So dentine is present between enamel and pulp area. It is secreted by odontoblast cells. The formation of dentine is known as dentinogenesis. It is a yellow-hued material, porous. This is the composition. And if a cavity is formed in the enamel and it continues up till dentine, then it leads to sensitivity, and this is the alive portion. Enamel was dead portion, whereas dentine is the live portion. Next, let's talk about pulp area. The dental pulp is a central part of the tooth that is filled with soft connective tissue. This area is the pulp chamber. As you can see, pulp chamber. It has blood vessels and nerves. This tissue contains blood vessels and nerves that enter the tooth from the hole at the apex of the root. So this is basically the apex. This is the root area, right? This is root area. So apex of the root is this area. So blood vessels and all travels from a hole. This is the hole, right? This is the hole, and from here the blood vessels enter into the pulp chamber. As you can see here, various blood vessels are there. Along the border between the dentine and the pulp are odontoblast cells, which initiate the formation of dentine. Other cells include fibroblasts, preodontoblasts, macrophages, T lymphocytes. The pulp is commonly known as the nerve of the tooth. So till now we know that pulp is a central part. It is filled with blood vessels and nerves. It has cells like odontoblasts, fibroblasts, macrophages, etc. and it is commonly known as the nerve of the tooth. Then if you talk about the cementum, then cementum is basically a layer of connective tissue that binds the root of the teeth firmly to the gums of the jawbone. So the root is bound to the gums and the jawbones through this connective tissue known as the cementum. Here you can see this is the cementum. Its coloration is yellowish and it is softer than dentine and enamel. I missed a point here. Cementum is bone-like substance that is covering the root of the tooth. So it is like the outer portion of the tooth. So cementum is basically covering the root area and then binding that root area to the gums and to the jawbone. The principal role of cementum is to serve as a medium by which the periodontal ligaments can attach to the tooth for stability. So basically what cementum is doing is cementum is binding different structures together. It is binding the root with the gums and the jawbone for the purpose of stability, so that our tooth can be stable, right? Then there are some other supporting structures also, that is the periodontal ligaments. Periodontal ligament is a specialized connective tissue that is responsible for attaching the cementum of the tooth to the alveolar bone or the jawbone. This tissue covers the root of the tooth within the bone. Then there comes alveolar bone. It is the bone of the jaw which forms the alveolus around the teeth. This is basically the jawbone. Like any other bone in the human body, alveolar bone is also modified throughout the life. It is formed and it is destroyed. Osteoblast cells create bone, whereas osteoclast cell destroys it. Alveolus is also known as bony socket. It is the bone of the tooth socket in which all the teeth are embedded. The gingiva or the gum is the mucosal tissue that overlays the jaws. You can see the pinkish reddish portion that you can see from your eyes. That is basically the gingiva or the gums. 1/3 of the crown is covered with the gums. So I hope you understood the different parts of human teeth. First of all, it is divided into crown, neck and root. Then there are different structures like enamel, dentine, pulp, cementum, then supporting structures like periodontal ligament, alveolar bone, gums, right? All the important points I've discussed in this video, you must remember them all. Two types of teeth, that is primary teeth and permanent teeth. Primary teeth have many different names, like for example, it is also known as milk teeth, or it is also known as deciduous teeth, or it is also known as temporary teeth, right? There are different names that are given for primary teeth, so we'll be discussing about these. So let's get started. First, I'm going to give a small description about human dentition. Basically, there are 4 types of teeth, that is incisors, canines, premolars and molars, and they have different functions. They have been discussed by me in one of the video in detail, all the important points that you need to know about incisors, canines, premolars and molars, like the number of roots they have, the number of cusps they have, their functions and all. The link for that video is given in the description box. You can go check out that video after this video if you want. Like for example, the incisors, they cut the food because their edges are very sharp. Canines are used for tearing the food. Molars and premolars are used for crushing the food. All these points have been discussed in detail in that video. You can go check that out. So, the roots of the teeth are embedded in the maxilla, that is the upper jaw, or the mandible, that is the lower jaw, and they are covered by gums. So if you look at this picture, the upper teeth, they are known as the maxillary teeth. As you can see here, they are the maxillary teeth, whereas the lower teeth, they are the mandibular teeth. The teeth that you can see from the front are the anterior teeth, and the ones that are present at the back are the posterior teeth. Humans are diphyodont, thecodont and heterodont. Now what are all these 3 terms? These 3 terms along with many other terms have also been discussed in a separate video. Link for that also is given in the description box, but for now I'll tell you what these 3 are. Diphyodont means two different types of teeth are present in the human dentition in different stages of life. Like for example, we know that there are first the primary teeth come, right? Then they go off, and then the permanent or the secondary teeth come, right? So die means two, so there are two types of teeth that comes in different stage of life. Then comes thecodont. Thecodont means that the teeth, they are embedded in bony sockets. Bony sockets. Teeth embedded in bony sockets. And heterodont, hetero means many types, so heterodont means that there are many types of teeth, that like we have incisors, canines, premolars and molars, right? If you want to have more information about these terms and few other terms, do check out that video. Now let's talk about primary teeth, because they are the teeth that are formed first, right? So they are also known as temporary teeth, baby teeth, deciduous teeth, milk teeth. So these are the different terms that are given to primary teeth. The dental formula, this is important for primary teeth, is 2102 by 2102. 2102 by 2102. And similarly 2102 by 2102. So it has to go like this. Okay, this is this is for the upper jaw, and this is for the lower jaw. That is, this is the maxillary teeth and this is the mandibular teeth. All right, so you need to remember, you can remember for half portion, right? 2102, 2102. So in 2102, the first one, let me just write this again like this. 2102, the first number is denoting the incisors, second number is denoting the canines, third is denoting the premolars, and fourth is denoting the molars. All right, so there are two incisors in the first quadrant, two incisors in the second, two third, and two in third and fourth. So there are a total of 1 2 3 4 incisors. Okay, then there is 1 1 1 1 canine. So I'm so sorry, 1 2 3 4 5 7 8 incisors, and there are 4 canines, and we have 0 premolars, and we have 8 molars. So in total we have 8 plus 4 plus 8 teeth in primary dentition, which is equal to 20 teeth. Alright, among these 10, talking just about one jaw, we have a maxillary, that is the upper jaw, and then the rest 10 are found in the mandible, that is the lower jaw, and there are a total of 20 teeth. In the primary set of teeth, there are two types of incisors, that is central incisors and lateral incisors, and there are two types of molars, that is first molar and second molar. All primary teeth are normally later replaced by their permanent counterparts. So I hope you understood everything about the primary teeth. Now let's move on to permanent teeth. So the dental formula for permanent teeth is 2123 by 2123. Similarly 2123 by 2123. As I told that this number indicates incisors, this indicates canine, this premolar, and this molar. So there are 8 incisors, 1 2 3 4, 4 canines, 1 2 3 4, that is 4 into 2, 8 premolars and 12 molars. So generally there has to be 32 teeth in all, 16 in the maxillary jaw and 16 in the mandible jaw, but third molar might or might not come, so it's not mandatory that each and every person is going to have 32 teeth. All right, so the maxillary teeth are maxillary central incisors, that is this one, maxillary central incisors. Then comes the maxillary lateral incisors, that is these maxillary lateral incisors. Then maxillary canines, that is these maxillary canines, maxillary first premolars, maxillary first premolars, maxillary second premolars. Here are the maxillary second premolar. Then comes maxillary first molar, maxillary second molar, and maxillary third molar. First, second, and third molars. Similarly, there are the mandibular teeth. These are the central incisors, lateral incisors, canines, first premolar, second premolar, first molar, second molar, and third molar. Similarly, it goes like this for here. The third molars are commonly called wisdom teeth, and they may never even erupt in the mouth or form at all. The age between they are generally formed is 17 to 25. So I hope you understood what permanent teeth is, what is the formula of permanent teeth, where are the different teeth located. Now let's just look at a few differences between them. The temporary teeth are smaller, narrower, and more delicate than the permanent teeth. The permanent teeth are heavier, bigger, stronger. If you look at the color, then the temporary teeth are white, whereas permanent ones are ivory. In temporary teeth, the neck area is more constricted, whereas in permanent teeth, the neck area is less constricted. Tooth edges are serrated in temporary teeth. They are not serrated in permanent teeth. Neonatal lines can be seen in temporary teeth. Neonatal lines here in this case cannot be seen except in case of first molar. The dentine is less mineralized in case of temporary teeth, and it is more mineralized in case of permanent teeth. So these are few of the differences between temporary and permanent teeth. We understood what primary teeth is. The dental formula is 2102. The permanent teeth has dental formula of 2123. Here are a few differences. So this was about a few dental terminologies like what is homodont, heterodont, monophyodont, diphyodont, polyphyodont, acrodont, pleurodont, thecodont. What are all these terms? So that will be discussed in this video. Also, if you want this PPT, then I have uploaded this on my website. You can go check out my website. The link for website is given in the description box. So now let's get started. So dentition pertains to the development of teeth and their arrangement in the mouth. In particular, it is the characteristic arrangement, kind, and number of teeth in a given species at a given age. So whenever we see the term dentition, what comes in our mind is teeth, right? Dentition is related to the development of our teeth, the arrangement of our teeth, how the teeth are arranged in our mouth, what kinds of teeth are there, how many teeth are there, that is the number of teeth present at a particular age or in a given species. Also, morphophysiology of the teeth is also studied in dentition. Morphophysiology means the relationship between the shape and form, that is the morphology, how the teeth looks, its shape and form, and its function, that is the physiology. So first let's start with the two terms, that is homodont and heterodont. As you can see in homodont, there is a word called homo. Homo means single. So the homodont are those organisms that have all same type of teeth. Only one type of teeth is present in homodont, and the heterodont are those organisms that have different teeth. So here we have heterodont. Heterodont means many. So the heterodont includes many types of teeth, like for example incisors, canines, premolars, and molars. But the homodonts do not show any such morphological differences in teeth. Homodont have single type of teeth. Heterodont has many different types of teeth. The example of homodont is non-mammalian vertebrates. So all the vertebrates that are not mammals are homodont. That is, they have only one kind of teeth. Whereas heterodont example is humans. You know that we have 4 different types of teeth: incisor, canine, premolars, and molars. So our teeth are heterodont. If you look at this picture, This is a reptile versus mammalian dentition. This here you can see is a homodont dentition. All the teeth are only of one kind, that is seen in reptiles, whereas here we have heterodont, that is we have different types of teeth: canines, incisors, premolars and molars. This is a picture of a dolphin skull. Here you can see all the teeth are exactly the same. Only one kind of teeth are present in here. So this dentition is homodont dentition. Now let's move on to our next terms, that is monophyodont, diphyodont and polyphyodont. Here you can see mono. Mono means one. So, a monophyodont is an animal with only one set of teeth that grows continuously throughout its life. So, in monophyodont, we get to see only one set of teeth throughout the life of that animal. The example includes platypus, sloth, walruses, seals, narwhals, dolphins, and most toothed whales. Whereas di means two. So this means that two types of teeth occurs in the life cycle. Humans and most other mammals are diphyodont, which means that we have two successive sets of teeth. So we know that first our baby teeth come, right, which are also known as deciduous, primary or milk teeth, and after that our adult teeth or the permanent teeth or the secondary teeth come. So there are two types of teeth, hence human teeth are diphyodont. Then comes polyphyodont. Poly means many. So a polyphyodont is any animal whose teeth are replaced many times until their tooth bud completely depletes. So in monophyodont only one teeth come only once in life. In diphyodont teeth come twice in life. In polyphyodont the teeth are going to come many times until the tooth bud is completely depleted. Most vertebrates, tooth fishes, reptiles are polyphyodonts. The only mammalian polyphyodonts are manatees, kangaroos and elephants. Then the other three terms are acrodont, pleurodont and thecodont. Acrodont are the weakest and do not have a firm attachment inside the jaw. Here you can see that the teeth, this is the jaw portion, alright. This grey portion, the one that is being marked in red now, is the jaw portion. In acrodont you can see that it is just superficially attached on the surface of the jaw. It is not embedded, inside. It is not embedded. It is just touching the surface. There is no firm attachment inside the jaw. It is just attached on the surface. They are superficially attached to the jaw and aren't deep in the bone. They can easily be broken, of course, with enough force, because they are not embedded, they are just superficially attached. This tooth formation is seen in animals like chameleons, uromastices, frilled dragons and bearded dragons. The next term is pleurodont. Pleurodont teeth have longer roots with weak attachments to the mandible and no socket. Here you can see that the root area, this area is longer in pleurodont, but the attachment is still weak and there are no proper sockets. Pleurodont teeth will regenerate if lost, but the acrodont will not. So if due to some accident the pleurodont teeth is broken, then that teeth will be regenerated, but if this happens with acrodont, then there will be no regeneration. Then comes thecodont. Thecodont is a morphological arrangement in which the base of the teeth is completely enclosed in a deep socket of bone. Here you can see the root area is completely enclosed or embedded in the bony socket. So this happens in case of humans. So humans have a thecodont dentition. So till now we know that humans are, first of all, humans are heterodont. That means we have different types of teeth, that is incisors, canines, premolars and molars. Then human teeth are diphyodont, which means that our teeth comes, there are two different types of teeth that comes in different stages of life. And lastly, human teeth are thecodont, that is, these teeth are embedded in the bony sockets. Few methods by which we can determine the age of a person by using his or her teeth. So let's get started. Dental age assessment is one of the most reliable methods of chronological age estimation used for criminal, forensic, and anthropologic purposes. Teeth are known to aid in personal identification and age determination, as they are highly durable to resist the effect of putrefaction by chemical and other environmental factors. So teeth can help us know the age of a person even after death. Why? Because teeth are highly mineralized, and because of that reason they are durable and they resist putrefaction process. While other organs start to get putrefied by chemical and other environmental factors, our teeth are going to resist putrefaction, and hence they are going to stay the same. Administration of justice often requires estimation of age of an individual in litigations involving both civil and criminal cases, such as issues related to consent, employment, retirement, marriage, case of sexual offense, kidnapping, abduction, criminal responsibility, juvenility, etc. So teeth have been useful in estimating the age of a person in a number of cases like these. It is a very reliable method to determine the chronological age of a person. And for that we need great deal of visual examination along with subject expertise. So we need to be very much thorough with the knowledge. Other than the knowledge, we also need great deal of visual examination. The forensic odontologist must thoroughly study each and every aspect of that particular teeth in order to determine the age of the person, and hence visual examination is very important step in this. Now there are different methods for determining the age. So dental age estimation in child and in adolescent, there are three methods, that is Atlas method, which is a radiographic method, then there's Shore and Massler method, and then there's Demirjian method. Then for determining it in adults, we have Gustafson radiological method, and there are some other methods like visual method, amino acid racemization also. So in this video I'm going to be discussing about these three methods, that is the methods that are used for age estimation in children, and all the other methods will be discussed in my next video. So let's start with the Atlas approach. Radiographs. In this method, radiographs are used to morphologically assess the distinct stages of mineralization of tooth. A radiograph is basically a static image that is generated when X-rays are passed through the patient's teeth, in case teeth. So X-rays are used in this method and radiographs are made. This is a radiograph. This picture is a radiograph of teeth, and with the help of these, the odontologists are going to study the various stages. There are various stages of mineralization of teeth. So which stage the person's teeth were in, we can determine that, and on the basis of that, age of the person can be estimated. The tooth mineralization stages are much less affected by variation in nutritional and endocrine status, unlike bones, hence provide a more accurate indication of chronological age. So in cases of bones are also mineralized, teeth are also mineralized, but the bones mineralization stages can be affected by the nutritional habits of the person or the endocrine status, and hence it can we cannot determine the age through that. But this is not so in teeth's case. Teeth mineralization is not affected by this by nutrition or by endocrine status, hence it is more reliable. The advantages of Atlas method is dental radiographs are non-destructible. Of course, these radiographs cannot be destructed. Also, the X-rays don't affect the teeth in any way. So dental radiographs can be used readily and reliably. Dental radiographs are of enormous value to determine the age when only skull is found and other body parts or long bones are not available for the purpose of estimation. So when only skull is found, then dental radiographs become even more important. because we don't have any other part that we can look for age estimation. So in case of when only skull is present available, then this becomes even more important. Then the second method is Shore and Maslow's method. So Shore and Maslow basically describe 20 different chronological stages of dental development, starting from 4-month age after birth to 21 years of age. Here is a picture that is showing all those 20 stages. The individual dental status was compared with this table to estimate age. So here in this picture you can see basically blue colored, they are showing us the deciduous dentition of the primary teeth, whereas this yellowish part is showing us the permanent teeth, right? First we have deciduous teeth, then we have mixed dentition, that is a mix of deciduous and permanent teeth, then comes the finally permanent teeth. So in different stages, some of the teeth are erupting, some are still inside the jaw, like this is inside the jaw, these are erupting. So there are various stages, and if we have a set of teeth we need to estimate the person's age, then what we can do is we can study those teeth, we can see how many have erupted, how many are still inside, and on the basis of that we can, we have the estimated age written over here in this chart. So the individual dental status can be compared with this table to estimate the age. Then we have Demirjian's technique. The development of each permanent mandibular tooth except for the third molar was rated on 8 stages from A to H. So basically Demirjian, he rated the teeth on 8 scale that started from A to H, A, B, C, D, E, F, G and H. Each letter corresponds to a score. The sum score of all the 7 teeth represents the dental maturity score. And these scales means different stages, like for example A here means mineralized cusp tips not yet coalesced, B means mineral cusps united, C means the crown is approximately half formed, D means crown formation is complete to dento-enamel junction, E means root formation has become, F means root length at least as great as crown length, G means parallel root walls with open apices, H means apices are completely closed. So these are various stages that are given. So we have this, then we have the teeth that we have to estimate the age of. We are going to sum the scores of all the 7 teeth, and then we are going to get a dental maturity score. The dental maturity score can be converted directly into dental age by using a graph or a table of standards. So I hope you understood these 3 techniques. Atlas approach used radiographs, and then the mineralization stages were studied. In Shore and Maslow, there were 20 chronological stages that were representing the teeth development, which can be used to compare the individual dental status of a person. Then Demirjian's technique rated different teeth from A to H, and then those scores were summed and the dental maturity score was converted then into dental age. Method called Gustafson's method, which is basically a method for calculating the dental age of a person on the basis of 6 criteria, that is adhesion, periodontosis, secondary dentine, cementum apposition, root resorption and transparency of the root. All these 6 criteria will be discussed in this video, along with a question on how we can actually use this Gustafson method to calculate the estimated age of a person. So let's get started. Gustafson studied the changes occurring in an individual teeth and succeeded in estimating the age with some accuracy. The following 6 changes were studied for age estimation. So in the year 1950, Gustafson came up with a method for estimating the age of a person using the teeth. He proposed 6 criteria on the changes that occurs in the teeth with age, and he developed a ranking scale. A ranking scale was developed by him. The scale goes from 0 to 3, that is 0, 1, 2 and 3. And the 6 criteria that he used are atresion, periodontosis, secondary dentine, cementum apposition, root resorption and transparency of root. Now let's study these 6 criteria one by one. First is atresion of teeth. What is atresion? Atresion means physiological wear and tear of occlusal surfaces of the teeth due to mastication. Now our teeth are responsible for a number of reasons like grinding, chewing, tearing, etc. Right? So with time what happens is that the wear and tear of teeth starts to occur, due to which atresion occurs. This you can see over here is a healthy teeth, whereas this is a teeth in which atresion has occurred. The teeth was supposed to be till here, right, as you can see, but due to its wear and tear it has decreased in its length, right? So we have 4 scales for atresion, that is A0, A1, A2 and A3. As I said, in this method the 6 criteria have been ranked on a scale of 0, 1, 2 and 3. So here we have A0. A is because we are studying the criteria of atresion, so A is used. A0 means there is no atresion. A1 means that atresion is in the enamel area. A2 means atresion has reached the dentine. A3, most severe one, means that atresion has reached the pulp area. Then the second criteria is secondary dentine. Secondary dentine is a layer of dentine which is produced after the tooth's root is completely formed. So with age, so normally we have only one dentine, but with age what happens is a secondary dentine starts to get formed. Here you can see in this picture, this area is the primary dentine area. But with age what happens is we get to see a secondary dentine area also. This area here you can see, this is the secondary dentine area, alright. So the 4 scales are S, there is a mistake here, this has to be S0, S1, S2 and S3, okay. S0 means that there is no formation of secondary dentine. S1 means the secondary dentine has started to begin in the pulp area. S2 means pulp cavity has half filled with secondary dentine. S3 means pulp cavity is full filled with secondary dentine. So these are the 4 scales of secondary dentine criteria. Now let's talk about the third criteria, that is periodontosis. Periodontosis basically is the inflammation of periodontal tissues or periodontal ligaments or gingiva with age, that is leading to loosening of the teeth in the socket. So basically in periodontosis, the gingiva, the gums or the periodontal ligaments, they are getting inflamed, right, due to which what happens is that the teeth starts to become loosened in the socket. There's no proper grip here. If you can see in this picture, this is a normal tooth and this is a tooth having periodontosis. Here we have this red area that you can see, this is the gum area, alright, and here we have the healthy gums. But in case of periodontosis, we can see that the gum area has displaced from its normal position. There's a gap that you can see over here. This gap is due to the inflammation of gingiva or the periodontal ligament. So this is what happens in periodontosis. Here we have 4 scales, P0, P1, P2 and P3. P0 means there's no periodontosis. P1 means that slight loosening of teeth has started. P2 means periodontosis along 1/3 of the root has started, and P3 means periodontosis along upper 2/3 of the root has happened. So basically, how are we going to use this method? We are going to see the teeth of the person. We are going to see how much atresion has occurred. Has the atresion just reached the enamel or has it even reached the pulp? If it has reached the pulp, to have the score of A equals to 3. Then we'll look for dentine. Is there any dentine formation is a secondary dentine formation? No, there is no. If there is no, then S value will be 0. And later on we are going to calculate for all of these criteria we are going to calculate, and then we have a formula at the end on the basis of which we are going to calculate the entire age of the person. Alright, so coming back to the fourth criteria, that is the cementum apposition. That means the deposition of cementum around the root of the tooth. So C0 means that there is no deposition of cementum. C1 means apposition is slightly greater than normal. C2 means there is greater layer of cementum, and C3 means there is very heavy layer of cementum. Alright, then next criteria, the fifth criteria, is the root resorption. Root resorption is the progressive loss of dentine and cementum by the action of odontoclast cells. So we know that there are two types of cells, that is odontoblast cells and odontoclast cells. Odontoblast cells are responsible for the formation of tooth cells, okay, whereas odontoclast cells lead to degradation of the dental cells. Odontoclast cells are basically responsible for the resorption of the root of the deciduous teeth. So root resorption basically means that there is loss of dentine and cementum. Here you can see in this area the root, in this radiograph over this area you can see that the root resorption has been shown. So R0 means there is no root resorption. R1 means only small isolated spots of resorption can be seen. R2 means lower 1/3 root resorption is there, and R3 means that there is greater area of root resorption. Then the last criteria is the transparency of root. With age, the dental tubules are filled with minerals and they start to turn opaque, due to which the root starts to become slight transparent. This is the most reliable criteria of the all. This is 0 score, that is T0 score. Then here comes T1 score, that there is slight transparency. Then here we have T2, which means transparency is over 1/3 of the apical root, and T3 means the transparency is over 2/3 apical root, which will also include transparency in the whole root. So these are the 6 criteria that are used in the Gustafson's method: atresion, secondary dentine, periodontosis, cementum apposition, root resorption, and transparency of the root. Now let's see the formula that is used for calculating the age in Gustafson's method. So the age estimation can be done by totaling the scores. Total score, how we are going to calculate: A score plus P score plus R score plus T score plus C score plus N score, that is atresion score, periodontitis score, root resorption score, transparency of root score, cementum apposition score, and secondary dentine score. Alright, these scores has to be added up, and this score is to be kept over here in this formula, that is in the total score area. So basically the formula that is used is 11.43 plus 4.56 into the total score that we will be getting from here after the proper examination of the tooth, plus minus 3.63 years. So let's try to find out with the help of an example. If we have studied a teeth, then we are going to have all these values of A, and for P, for R, for T, for C, and for N. We'll be having these values, right? What does the value means? Like in this question we have the values A equals to 1. What does A equals to 1 means? A equals to 1 means that atresion is in the enamel. So we studied the teeth and we found out that atresion has only reached the enamel portion. So we deduce that A equals to 1. Similarly, we have here P equals to 0. P equals to 0 means that there is no periodontitis, no inflammation, right? So accordingly, we have been given these values, but we just have the values. How can we calculate the age of the person? So we'll be doing that with the help of this formula. Remember this formula. So if these values are given, so let's find out the age of the person, which is going to be 11.43 plus 4.56 into the total score. What will be the total score? Total score will be 1 plus 0 plus 1 plus 0 plus 1 plus 1, right? 1 plus 0, 1 plus 0 plus 1 plus 1, right? Then plus minus 3.63 years. So 11.43 plus 4.56 into 1, 2, 3, 4 plus minus 3.63 years. 11.43 plus 4.56 into 4 is 18.24 plus minus 3.63 years. So 18.24 plus 11.43 is equal to 29.67 plus minus 3.63 years. So just deduct this 3.63 and add this 3.63 into this term to get a range. So 29.67 plus 3.63 is 33.3 years, and 29.67 minus 3.63 is 26.04 years. So through this method we have reduced that the age of the person can be between 33.3 years and to 26.04 years. It will be between this age. Alright, so I hope you understood how to calculate the range by this method. Now I have a second question for you guys. Find out the age of the person and let me know the age of the person in the comments. Dental age estimation methods that were left. In my previous video I've already discussed these three methods, that is Atlas method, Schoren-Massler method and Demirjian's method. These are methods that are used for age estimation, dental age estimation in children and in adolescents. In today's video I'll be discussing about these three: visual, Gustafson and amino acid. Gustafson, I've already made a separate video for Gustafson since it is quite a big method, so that video's link is given in the description box. Let's talk about the other two methods now. First is the most basic or the simplest one, that is visual method, in which simply we visualize the teeth carefully and we study the teeth and then we try to give an estimate age of that person. So this is the simplest age estimation method. This method is based on clinical expertise without using any formal method. So we need clinical expertise, we need the knowledge about the subject, that is the knowledge of odontology is required. With knowledge what we also need is experience, right? So for this method the forensic odontologists need to have the subject knowledge and as well as the experience is required. And of course this method is questionable because how we perceive something is subject to questions. How another person is going to perceive the same thing can be different. So this method is questionable. Then Gustafson's method, which is very important method, has already been discussed. In short, I can discuss it here. Like Gustafson in 1950, he came up with a method in which 6 dental changes were studied, which were adhesion, periodontitis, secondary dentine, cementum deposition, root resorption and transparency of root. And these 6 criteria or changes, we can say, they were ranked basically from 0 to 3. 0, 1, 2 and 3. 3 ranks were given. Like for example, adhesion, A is the symbol for adhesion, and A0 means there was no adhesion. A1 means that adhesion is there in the enamel. A2 means adhesion is there in the dentine. A3 means adhesion has reached the pulp area. So all these changes, dental changes, they are scaled, right? So you can check that video out. I have explained the entire Gustafson method very clearly in that particular video. Then talking about the next method, that is amino acid racemization. So the relationship between the extent of aspartic acid racemization in dentinal biopsy specimens and age is very close, and it facilitates age estimation. Now, as the age advances, as we grow old, the L-aspartic acid is an amino acid. So this aspartic acid, L-aspartic acid, starts to get changed into D form, D-aspartic acid. So L-aspartic acid starts to get converted into D-aspartic acid, which is called racemization. There are a number of changes that occurs in the proteins, a number of chemical reactions like oxidation, isomerization, racemization. These changes occur, these are age related changes that occur. Among these changes, one is racemization. Racemization means conversion of L form to D form, and this starts to occur as the age progresses. Now in living body, normally what happens is the proteins that are synthesized, the newly formed proteins, are in L form. Okay, the newly formed ones are in L form, although there are some exceptions, but most of them are L form, and with time racemization occurs and they gets converted into D form by some chemical reactions. So by calculating the amount of D-aspartic acid that is present in the body, we can give an estimate on how old the person is, because if there is more amount of D-aspartic acid than L, the person is old, whereas if more amount of L-aspartic acid is present, that means the person is young. So this method is exclusively used in age estimation for unidentified corpse. The method involves extraction of one or more teeth. Hence, this method was not applied on living humans due to ethical concern. Of course, since the method needs one or more teeth to be completely removed or extracted, therefore this is not applied on living humans. Therefore, a special biopsy technique applicable to dentine was developed, which caused only minor discomfort to the subject. This is the formula that is used to determine the coefficient of racemization, in which A is rate constant of aspartic acid racemization, whereas B equals to y-intercept. And D and I are integrated peak areas of the respective enantiomers. A is, okay, it's written over here, A is the rate constant of racemization of aspartic acid in dentine, and B is y-intercept. So I hope you understood the methods that are used for dental age estimation in adults. First is the visual method, and then the most important one is the Gustafson method, which has already been discussed in a previous video. You can go check that video out. And then there is amino acid racemization method. The eruption sequence of teeth, that is both primary as well as permanent teeth. So let's get started. First, let's talk about the eruption sequence of primary teeth. So we all know the dental formula for primary teeth. It is 2102. Similarly, 2102 for this quadrant, 2102 for this quadrant, and 2102 for this quadrant. So we will be discussing just about these two quadrants, because the eruption sequence is exactly same for this quadrant also. So let's discuss this one. This is drawn over here, right? So to remember the eruption sequence of primary teeth, you have to remember IMCM. This means that first of all incisors erupt. And which incisors? The mandibular incisors. First there are central incisors, then there is lateral incisors, then there are canines. There are no premolars in primary teeth, so there's first molar and there's second molar. Similarly, we have central incisors, lateral incisors, canines, first molar and second molar. So if according to this formula IMCM, first tooth that erupts is the incisors. Now we have 4 incisors over here, right? So which teeth is the one that erupts first? So it is the central incisor in the mandibular jaw, that is the lower jaw, that erupts first. Alright, as you can see over here also, 1 is written over here. Then erupts the central incisor in the maxillary jaw, as you can see here. Then the lateral incisors in the maxillary jaw erupts, and then the lateral incisor in the mandibular jaw erupts. Then in this formula we have M, that is the molar is going to erupt. Which molar? The first molar. Here we have, this is central incisors, this is lateral incisors, this is canine, this is first molar, this is second molar. So after the eruption of this, that is lateral incisors in the lower jaw, the next teeth is molar. And which molar? Upper molar first. So 5 and then 6. Next teeth is going to be canine. So 7 and 8. And the last ones are going to be the second molars, but the lower jaw first. So 9 and 10. So this is how the teeth gets erupted. You need to remember this formula, IMCM. So now let's talk about the eruption time of these teeth. So let me just write down. First, the central incisors come, the lower one. Then the central incisors come, the upper one. Then comes the lateral incisors, upper one. And then comes the lateral incisors, lower one. Then comes the first molar, canines, and the second molar, right? This is what we have learned till now. Now let's learn the eruption time for all these teeth. Central incisors come in between 6 to 8 months. Central incisors, the lower one. Central incisors in the upper teeth, that is the maxillary teeth, comes between 7 to 9 months. Lateral incisors in the upper teeth comes in between the same time period, that is between 7 to 9 months. Then the lower incisor, the lateral incisor in the lower jaw comes in between 10 to 12 months. Then the first molar comes in between 12 to 14 months. The second canines comes in between 17 to 18 months. And finally the second molar comes in between the 20 to 30 months age. So if you look at this, on an average, a child is supposed to have 8 teeth in the first year age, and the child is supposed to have 18 teeth in the 1.5 year age, and the child is supposed to have 20 teeth in 2 to 2.5. Now let's talk about the eruption sequence of permanent teeth. For this you can remember this one line, that is, mama is ill, papa can make meal. So you need to remember the first alphabets here, MII, PP, CMM, and everything is sorted. This is the eruption sequence of permanent teeth. First, the first molars come, then the incisors, central incisors, and then lateral incisors come. Then the premolars come, first premolar, second premolar, then the canine, then the molar and molar. This is the eruption sequence of teeth. So the first molars erupt at the age of 6 to 7 years. The central incisors erupt at the age of 6 to 8 years. The lateral incisors erupt at the age of 8 to 9 years. The first premolars erupt at the age of 9 to 11 years. The second premolars erupt at the age of 10 to 12 years. The canines starts to erupt at the age of 11 to 12 years. The second molar starts to erupt at the age of 12 to 14 years. And lastly, the wisdom tooth, that is the third molar, erupts in between the age of 17 to 25 years, and in some cases it does not erupt at all. Tooth numbering systems, there are various systems like universal system, Palmer system, Haderup, FDI, modified FDI. So all these numbering systems, tooth numbering systems, will be discussed in this video. So let's start. Now in tooth numbering system, numbers starting from 1 to 32 along with alphabets and some unique symbols are used to identify the teeth in each quadrant. You know that there are 4 quadrants, right? First, second, third, fourth quadrant, right? And there are teeth in these quadrants, right? So to identify these teeth in each quadrant, we use the tooth numbering system. Now the question arises, why do we need this system? So the answer to that is that these systems helps to prevent errors when differentiating between right and left side of the mouth or upper and lower side of the mouth. So basically, to prevent errors or to prevent any kind of confusion so as to what teeth is the person referring to, these systems have been developed. These are fixed notations so that there is no confusion left and people can easily differentiate between different teeth. It also has advantage when typing and is capable of being incorporated in computer languages. Now there are several systems in the world, but the most widely used ones are universal system, Palmer system, which is also known as Sigmondi system, Haderup FDI, modified FBI, out of which this and this is very important. These are the most widely used systems, out of which FDI is most widely used among these 3 also. So first let's study about the universal system or the Cunningham system. Now the universal numbering system is also known as American system. Why? Because it is a dental notation that is used in United States. So that is why it is also known as American system. Now here we have the Cunningham system for permanent dentition and for primary dentition. So first let's talk about the permanent dentition. So always remember that the patient's right appears on the left side of the chart. So if this is the chart for permanent dentition for universal system, then the left area of the chart, that is this area, right? This is the left area, that is actually the patient's right. So the patient's right side appears on the left side of the chart. So hence we have right here and here we have left, and the patient's left side is actually on the right side of the chart. Alright, same goes with this chart also. This is right and this is left. The labels right and the left on the charts are actually corresponding to the patient's left and right and not on the person who is viewing. So in Cunningham system, both alphabets and numbers are used. For permanent dentition we use numbers and for primary dentition we use alphabets. The uppercase alphabet A through T are used for primary dentition. If we talk about the primary dentition or the deciduous dentition or the teeth that arrive first, then there are alphabets used ranging from A to T, whereas for permanent dentition numbers are used ranging from 1 to 32. And how does it start? The numbering starts from the upper left corner. It goes like this: 1, 2, 3, 4, 5, 6, 7, 8. This is the first quadrant, it is finished. Then the second quadrant start 9, 10, 11, 12, 13, 14, 15, 16. And after that we move downwards 17, 18, 19, 20, 21. Like this it goes. So first we go like this, then we go like this, and then we go like this. So this is like a clockwise direction, right? And similarly here in the primary dentition also, it goes like this: A, B, C, D, E, F, G, H, I, J, then K, and then like this. So here also we have a clockwise direction movement. So what you need to remember is that in universal system, in permanent dentition, the numbers are used, whereas in primary dentition, the alphabets are used, and the numbers and alphabets have to go in a clockwise direction starting from the upper right corner. Alright, now let's talk about the second system, that is the Palmer notation or the Sigmondi system. There are two names used for this, so don't get confused, it is the same thing. The Palmer notation method consists of symbols. These four symbols are used which designate the quadrant of the tooth, and also numbers are used which indicate the position of the tooth from the midline. So let's have a look here. First, the permanent teeth. So here in Palmer notation, first symbols as well as numbers are used. Symbol that is used, you know that this is a quadrant, right? So for this quadrant, this symbol is used. For this quadrant, this symbol is used. For this quadrant, this symbol is used. And for this one, this is used. So this is pretty easy, right? And the numbering here does not start from the upper right corner. It does not go straight. It starts from the midline. If this is the quadrant, then we know that this is the middle line. This is called the midline. So the numbering is going to start from this middle line only, right? So it is going to go like this: 1 2 3 4 5 6 7 8, then like this: 1 2 3 4 5 6 7 8. Similarly 1 2 3 4 5 6 7 8. So it goes like this, and we have these symbols, these four symbols to differentiate the quadrants. Adult teeth are numbered from 1 to 8, while primary teeth or the baby teeth, here if we look, they are indicated by the alphabets and not numbers. So it goes from A, B, C, D, E, A, B, C, D, E. I hope you know that there are 8 teeth in each quadrant, right? And what are those 8 teeth? The teeth are central incisors, lateral incisors, canines, first premolars, second premolars, first molar, second molar, and third molar. So how many are these? 1 2 3 4 5 6 7 8. So there are 8 teeth in each quadrant. So we have 8 teeth here, 8 here, 8 here, and 8 here. Whereas in case of deciduous teeth or the primary teeth, we only have 5 teeth. And what are those 5 teeth? We have central incisors, lateral incisors, we have canines, but there are no premolars, right? And then we have the first molar and the second molar. So there are only 5 teeth in case of deciduous teeth. So we have only 5 here, and here we have a total of 8 teeth. So I hope you understood the Palmer's system also. Now let's move on to Haderup system, which is very much similar to the Palmer notation, except for only one change, that a plus and minus sign is used to designate the upper and lower. In Palmer system, we were using these symbols, right? But in Haderup system, we are going to use plus and minus sign. If you look at this picture, if we talk about the permanent teeth, we can see that similarly 1 to 8 teeth are going from here: 1 2 3 4 5 6 7 8. But instead of this sign, we are having this plus sign. Plus sign is used for the upper teeth. All the teeth in the upper portion are having the plus sign, and the ones in the lower jaw are denoted by a minus sign. The same case is here with the temporary teeth also, that is, a plus sign is used to denote upper teeth, and the minus sign is used in lower teeth. But here's a difference also. In Palmer notation, what we were using for temporary teeth was the alphabets, but here we do not use alphabets. For deciduous teeth, a 0 was additionally placed in front of the number. So everything is same as the permanent one, except for there's a 0 placed in front of all the numbers, right? So this is the only difference. This notation was adopted by Scandinavian countries. Now let's talk about the FDI system. FDI is 2-digit system, and the full form of FDI is Federation Dentaire Internationale, or also known as FDI, World Dental Federation notation system, is commonly used for numbering and naming of teeth throughout the world. The system uses two numbers, hence the name FDI, two-digit system, for location and naming of each tooth. Now the jaw is divided into four quadrants. We know that the first number refers to the quadrant of a tooth. So here if you see, this is our quadrant. So in this case, the upper right quadrant is the first quadrant. So this is upper right, then this is upper left, then this is lower left, and this is lower right. Alright, so in this case what we do is, this is a two-digit system, right? So there has to be two digits. So this digit is always the quadrant that the teeth is present in. So for all these tooth, the first digit is always going to be 1. For all these, the first digit is always going to be 4, right? 4, as you can see here. Similarly, 2 for this quadrant and 3 for this quadrant. So first thing is sorted. Now let's talk about the second one. So the second is same as the one that we studied in the Palmer notation. From the midline we just have to go towards the end, right? Like this. So 1, 2, 3, 4, 5, 6, 7, 8. This was the case for the permanent teeth, but for primary teeth what we have is everything is exactly the same, just that the first digit is going to change. Here we were having 1, 2, 3, 4 quadrants, whereas for primary teeth we are having 5, 6, 7, 8 quadrants. So this is the 5th quadrant, 6th quadrant, 7th quadrant and 8th quadrant. And similarly the second digit is going to be starting from the midline. We are going to 1, 2, 3, 4, 5. Similarly 1, 2, 3, 4, 5, like this only. So I hope you understood the FDI two-digit system also. Then there's a small modification system also available for FDI. In this, the tooth and the quadrant are designated by different numbers. The right quadrant is designated by 2 and 3 and the left quadrant is designated by 1 and 4. So earlier in FDI what we had was 1, 2, 3 and 4. These were the quadrants name, right? So, and this is the right quadrant, this is the left quadrant, right? This was the case. But in modified system what we are having is that the right quadrant are designated by 2 and 3. Here you can see they were designated by 1 and 4, but in modified they are designated by 2 and 3, right? This is the right, and for the left quadrants they are designated by 1 and 4. So in a way they are switched, right? They are the opposite. So this was for FDI and this is for modified FDI. Bite marks, introduction to what bite marks are and a few questions related to that will be discussed in this video. If you want this PPT, then you can go check out my website. I have provided the link for my website in the description box. I've uploaded this PPT along with all the other PPTs over there, so you can go check that out if you want. Now let's get started. A bite mark may be defined as a representative pattern left in an object or a tissue by the dental structures of an animal or a human. So basically, whenever human skin or any other animal skin is inserted in between the jaws or the dental structure or the teeth, and if the jaws are closed and the pressure is applied, then what we get to see is a bite mark. So bite mark is basically a representative pattern of the dental structure or the teeth of animals or humans, and it can be formed on any tissue, that is human or animal skin, or even on any inanimate object. Bite marks are either left on the victim. If it is on the victim, then they are left by the attacker, or bite marks can be left on the perpetrator. If it is on the perpetrator, then it is left by the victim of the attack, or they can also be present on any inanimate object that is found at the crime scene. Now, sexual assaults, fight, homicide, abusive incidents often result in biting injuries and the necessity to involve the forensic odontologist. So, bite marks can be found in all these cases, that is sexual assault, fights, homicides, abusives. Sometimes it may be necessary to differentiate a bite mark caused by human dentition and the one caused from the bite of an animal. The complexity of biting injuries and their analysis and interpretation makes them a great challenge even for the most experienced odontologists. The complexity, the minute differences, the odontologists have to look for those minute differences that are present in each and every jaw. So that interpretation, that analysis is quite difficult and it acts as a challenge for the odontologist. Now let's talk about the bite mark analysis. Bite mark analysis, which is an aspect of forensic odontology. Forensic odontology we know is the study of teeth, right? And forensic is the application of various sciences for providing justice. So the using of the knowledge of odontology, that is the knowledge of teeth, for solving any criminal case is known as forensic odontology. So bite mark analysis is an aspect of forensic odontology. Bite mark analysis is the process by which odontologists or dentists attempt to match the marks found at the crime scene with the dental impression of the suspect. So basically we get certain bite marks at the crime scene. There can be two types of bite marks that are used in the analysis, that is bite marks at the crime scene and bite marks on the suspects. We are going to have a list of suspects, right? So we have to look if any kind of bite mark is present on their body also, which can be actually made by the victim in order to protect themselves, right? So in bite mark analysis there are four main questions that has to be answered. First, is it a bite mark? The mark that you're seeing on the body or on the object, is that even a bite mark? We have to determine that. Then if yes, if it's a bite mark, is it human bite mark or is it some other animal's bite mark? Then if it is a human bite mark, then what to do next? And after that, last, are there any features in the bite mark that can be compared to the dentition of a potential biter? So all these questions have to be answered in this bite mark analysis. So first question was, is it a bite mark? So bite mark is generally oval or it can be circular in shape, containing tooth and arc marks. So if we get to see an oval or circular shape having tooth or arc type shape like this, as we can see here in this picture, then yes, that is a bite mark. Then the question arises, is it a human bite mark? Then the characteristics for human bite marks are that they usually comprise of two opposing facing U-shaped arches. As you can see here, these are two U-shaped arches. Now this is the normal case, but in some cases only one also might be present. There is a possibility that the other jaw was not in contact with the surface, it was over a cloth or any other object, and due to which only one of the jaw is able to form the bite mark. So generally there are two arches, U-shaped arches, which leads to formation of an oval or an elliptical shape. So human bite mark is oval or elliptical, that may be separated by open spaces. Here you can see, or as a ring of marks. They have elliptical or oval pattern containing tooth and arch marks. Central bruising is often present. Here you can see the central red area, and it is caused by compression, positive pressure of the solid tissue between the teeth. Human bite mark may present as diffuse or specific bruising, abrasions, lacerations through the complete avulsion of the tissue and often in combination. So human bite mark may have bruises, abrasions, lacerations, or even complete eversion. Complete skin has come out. That is also a possibility. Now we have a bite mark. Now what to do next? So early recognition is very important in bite mark cases. Why? Because bite marks will start healing themselves. Healing might occur and these marks may quickly fade or change in both living and dead. So we need early recognition and action. That is very necessary. So what we need to do is all these points will also be discussed in detail in my video of bite mark analysis. You can also go check that video out, but in short I'm explaining it here that first of all we have to do notes making. We have to describe how the bite marks are looking, where they are located, what is the shape and the color and the size and the type of injury that is caused. All these points have to be noted down. Then the collection occurs. Before that, photography is very important. As you can see here, a scale has to be used for photography. Scale has to be placed near the bite mark and then photograph has to be taken. The photograph has to be very clear, which it should be telling about the location of the bite mark, the shape, color, size, everything has to be captured in that photograph. After photography is done, saliva swabbing and impressions will be made. And similarly, impressions will also be made from the bite marks of the suspect, the list of suspects that we are having, and after that these bite marks that are found at the crime scene will be compared with the bite marks that we have got from the suspects. Then questions comes, are these features in the bite mark that can be compared to the dentition of a potential biter? Bite marks, like fingerprints, are unique to every single person. This means an investigator can tell a lot about the perpetrator or the biter, which can make the hunt for suspect easier. A gap in the bite mark, like for example, if there is a gap in the bite mark, that means that there is a chance that there is any tooth missing, right? And we have a suspect who is also missing a teeth, then we can be very much sure that okay, this person is the culprit over here, and that makes the search even more easier. These impressions can be matched against the dentition and dental impressions of the victim and suspect. So matching comparison has to be done. It has been established as an efficient tool in identifying criminals involving what all cases? In cases of abduction, child abuse, homicide and sexual assaults. So this was all about this introductory video about bite marks. In my next video, I'm going to discuss about the various types of bite marks, how bite mark analysis has to be done in a sequential manner, and lots more. It's about the classification of bite mark or the types of bite mark, and there are going to be 4 different categories of the classification, that is on the basis of appearance, on the basis of cause, then there's Cameron and Sins classification, and then there's Webster classification. Also, if you want this PPT, then I've uploaded it on my website. Link for the website is given in the description box. You can go check that out. Now let's get started. So first, on the basis of appearance, that is how the bite mark is looking like. So first comes evulsive bites. In evulsive bites, basically a section of the tissue or the skin is completely teared out of the body, leaving an ovoid gaping laceration. If you can see in this picture, this particular area, this has been completely teared out, right? The skin has been completely teared or detached from the body of the person. So this is what happens in evulsive bites. For example, in case of apple, when we eat an apple, we tear out a portion of apple from our teeth completely and then we eat it, and after that we get to see an ovoid gapping in that apple, right? So this is example of an evulsive bite. Then comes single arch bite. Occasionally, bite marks show only one arch. Now there can be a number of reasons for that. For example, superimposed clothing can be there, due to which the other half of the jaw is not direct in contact with the skin, hence only one arch is formed. Or uneven surface can be there, or the biting force is uneven, that is more force is applied from one jaw and little force is applied from the other. Or sometimes due to even absence of teeth, that is the teeth are not present and due to which only one arch is formed. If you look at this picture here, we can see only one arch is there, so this is an example of single arch bite. Then comes central contusion bite, in which we get to see an area of ecchymosis. Ecchymosis basically is reddening of the skin due to bleeding. There's bleeding under the skin, due to which the skin starts to look reddish in color. So area of ecchymosis is seen in the center of the bite mark. It is attributed to suction or tongue thrusting. Then fourth type is closed arch bite mark, that is both the arch, they are joined at the end. If you look at this picture, both these arches are joined at the end area, right? So on the basis of appearance, there are four types: avulsive, single arch, central contusion and closed arch bite mark. Now on the basis of cause, what caused the bite mark? So on the basis of that, bite marks can be of three types. They can be sexually oriented bite marks, they can be child abuse bite marks, or they can be self-inflicted bite marks. So first let's talk about sexually inflicted bite marks. These marks are most commonly encountered during sexual offenses. The injury displays central or peripheral suck marks and the marks of anterior teeth with good definition. So sexual oriented bite marks are seen in sexual assaults or sexual offenses. Here we get to see a central or peripheral suck marks can be seen, and also the anterior teeth marks can also be seen. Then comes the child abuse bite marks. In child abuse cases, either aggressive, that is anger bite marks, or sexually oriented types of bite mark are seen. Bite marks in children invariably symbolizes child abuse until it is proven otherwise. So bite marks in case of children are generally due to child abuse only, but the exception is only when it is proved that no, it is not due to child abuse, it is due to some other reason. Next comes self-inflicted bite marks, that is bite marks that are caused by the person himself. So mostly these are found in the forearms of children, caused by them. Sometimes arms may be pushed into the mouth of the child to make them stop crying, or due to intense pain, the children may bite themselves to overcome the suffering. So these are the reason because of which self-inflicted bite marks can be there. Also mentally retarded or psychologically disturbed people may also bite themselves. In some cases, self-inflicted bite marks are also seen in Nyhan syndrome, which is an X-linked syndrome, recessive syndrome, and it is a disease in which there is insensitivity to pain. The person cannot feel any kind of pain. So these were the three types of bite marks on the basis of cause, that is sexually oriented, child abuse and self-inflicted. Now let's talk about Cameron and Sin's classification. Cameron and Sin's classification is based on the types of agent producing the bite mark and the material exhibiting the bite mark, or material on which the bite mark is made. So agents can be of three types. The agent can be a human, the agent can be an animal like a dog, cat, etc., or the agent can be mechanical, that is any inanimate object. Now if you talk about the material on which the bite mark is formed, then that can be a body tissue or skin. It can be any food item like for example cheese, chocolate, apple, right? Or it can be any other item. Next come Webster's classification. It is based on the bite mark on food stuff. So it can be of three types: type 1, type 2 and type 3. In type 1, what kinds of food are included? The food items that are fractured readily with limited depth of penetration, for example chocolate. In type 2, food items that are fractured with considerable amount of pressure, then that comes under type 2. An example for this can be apple. And in type 3, all those foods come in which the complete penetration of food item with slide marks is there, like for example the bite marks on cheese. So these were the 3 types in Webster's classification. I hope you understood all these classifications, that is on the basis of appearance, on the basis of cause, Cameron and Sin's classification and Webster's classification. About certain guidelines for bite mark analysis, which will be including documentation, the sample collection techniques, etc. If you want this PPT, then I have uploaded it on my website. The link for website is given in the description box. You can go check that out if you want. Now let's get started. Bite mark analysis is basically supported by 2 assumptions. The first assumption is the uniqueness of human dentition when it comes to positioning of teeth in the jaw. The first assumption is saying that the teeth are uniquely positioned in the jaw, and second assumption says that whenever bite marks are made, the individuality of the teeth is completely replicated. A replica of that individuality is formed on the human skin. Bite mark analysis guidelines are considered to be dynamic and not static, and will be modified by time as significant development evolves. So bite mark analysis is not a rigid process, that is, it doesn't mean that the guidelines will never change. It can change with time, so it is dynamic, it is not static. It can be modified by time as the technology develops. So early recognition and action is very important in case of bite mark analysis. Why? Because healing may occur. The body might start to heal the bite marks. So what we need is early recognition and action. Once the recognition is made of the bite mark, or the bite mark may fade quickly or change in both living and dead. Hence the bite mark analysis should be done as fast as possible. There are 5 major steps. First is documentation, then comes evidence collection and preservation, then comes dental profiling of questioned and suspect samples, then comes comparison, and last is report the result. So first let's study documentation. In documentation, the first thing that is done is the description of bite mark. The bite mark has to be described. In case of both living victim or deceased victim, the odontologist should determine and record certain vital information, which is, first of all, the name of the victim is to be written, then the case number, the date of examination, the person to contact, the age of the victim, sex of the victim, and name of the examiner or examiners. So all these points to be noted down in documentation, that is, the description of bite mark has to be given. Then the location of bite mark is the second thing that comes in the documentation. While describing the location of the bite mark, the following points should be kept in mind. Location means where the bite mark is present, where it is located. So the following points should be kept in mind. First is that we have to describe the anatomical location of the bite mark, whether it is present on your hand. If on hand, then what position on the hand. If it is present on your abdomen, or it is on your thighs, forearms, whatever place it is, we have to give the anatomical location. Then describe the surface contour, that is, if it is flat, the surface is flat, or it is curved, or it is irregular. Then describe the tissue characteristics as you are able to see. Then underlying structures have to be mentioned, whether it is a bone, cartilage, muscle, fat. Then you have to tell whether the skin is relatively fixed or mobile in that place. Next in documentation comes the shape of the bite mark. What shape the bite mark is of. For example, it can be round, ovoid, crescent shaped, or irregular. So you have to mention the shape. Then also you have to mention the color. The color of the bite mark should also be noted. Then the size of the bite mark. Mark vertical and horizontal dimensions. Vertical and horizontal dimensions have to be measured, noted down, preferably in metric system. Then type of injury. It is also important to describe the type of injury inflicted while describing about the bite mark. The type of damage caused by the skin is also documented. So these 6 points, that is, first description of the bite mark, name of the victim and all those things, then location, where it is located, shape, color, size, and type of injury, all these points have to be mentioned under documentation. Then comes photography. Photography is very important step considering the bite marks will start getting healed. So what we need is we need to preserve those bite marks. So how we can do it? We can do that by taking photographs, right? So photography is very important step. For that we can use conventional camera equipments, that is the DSLRs, that are manufactured for visible light spectrum. Right, DSLRs are for capturing visible light and it is the most common methods of documenting the bite marks. Other than that, IR light and UV photography, that is IR photography and UV photography is also done in certain special cases. So IR light is used in photography of injured tissue due to some special properties when teeth or other objects crush the tissue that leads to subdermal hemorrhage. The extravasation of blood beneath the skin surface is captured using this IR photography. And for UV photography is used in bite mark documentation due to its ability to accentuate the surface details of disrupted skin. 3 orientation photographs of the area should be taken. The 3 photographs are going to be close-up photographs, mid-range photographs, and far-range photographs. So close-up, mid-range and far-range photographs has to be taken, 3 orientation photographs. The photographic resolution must be of very high quality. Photographs of marks must be taken with and also without scale in place, like this. This is scale over here, this, and the scale must be placed here, and then the photograph should be taken. Also photograph without scale should also be taken. Sketching must also be done. Other than photography and notes making, sketching of the bite mark also has to be done. Now for collection of bite marks, the method that we use is saliva swabbing method. It is suggested that whenever possible, saliva traces must be collected in accordance to recommendations of testing laboratory. So whenever possible, whenever you think that okay, saliva might be present in this particular evidence, that saliva must be taken, because if we get DNA, then catching the suspect will become very easy. So for that we use double swabbing technique. In double swabbing technique, what we do is we take a swab. This is a swab, and swabbing an area with a moist or cotton swab. So this is a cotton swab and it is moist, slightly moist or wet, and it is followed by swabbing the same area with a second. So first this swab is taken, a moist swab is taken, and it is rubbed over the area so that the area's saliva can get transferred to the swab, and then this swab is taken in a bottle and it is kept there. The upper portion is cut into half and it is thrown away, whereas the main portion is left into the bottle and then the bottle is closed. Then another swab is taken, and that swab is not wet, it is a dry cotton swab, thus absorbing any excess liquid that is left at the place. Both swabs are processed together, supposedly increasing the chances of DNA recovery compared to just using one wet swab. So I hope you understand that why double swabbing technique is used, because in double swabbing technique the chances of getting the DNA are increased as compared to just single swabbing method. Then after doing that, what we can do is we can make impressions. Impressions must be taken of the surface of the bite mark when it appears that this might deliver useful information. So this will depend upon the forensic odontologist whether he or she thinks that making impression might help us in further investigation. So for that, powder paste mix is taken. The paste that is taken is silica based, right? So silica based impression material is preferred as it flows on the crease and provides accurate detail. This silica based impression has the ability to go inside the crease area of the bite marks, hence providing us accurate details or better quality impression is made. So the impression that we are going to get is going to be a negative replica of the bite mark. So as you can see here in this pictures, these are mixtures that are poured over the bite mark area. As you can see, once it is dried, these are taken off and what we get to see is a negative replica of the bite mark. After that, this thing that is taken off, that is used to make another cast which is going to be a positive replica of the bite mark, that is exactly same replica of the bite mark of the person. After that, dental profiling is done, in which odontometric measurements of teeth is done. Each and every teeth is measured, the length, height, breadth, everything. Then we look out for certain anomalies or abnormalities. The position of the teeth is noted, or the angulation of the teeth, the angle at which the teeth are situated or located is noted. So all these things are done in dental profiling. Now once everything is done, the comparison is made. Samples are collected from both the suspect and the victim, and those samples are then compared with each other, and we see what are the similarities, what are the dissimilarities, and on the basis of knowledge of the forensic odontologist and on the basis of experience, the odontologist is going to give the result, that is whether the suspect sample matches with the victim's sample or not, and after that a report is made. So these are all the steps. That is, first documentation is done, which will be including description of bite mark, location, shape, color, size, type of injury. Then photography is done, sketching is done, then collection of bite mark is done, both from the suspect as well as from the victim, in which we use double swabbing technique. Then if required, impressions are made and dental profiling is done. Then comparison of the sample from suspect and victim is done, and then the results are reported. So this was all about the bite mark analysis process. I hope you understood everything. Dental anomalies. This is a part 1 video. All these dental anomalies will be discussed in this video. So if you want this PPT, then I've uploaded it on my website. You can go check that out. Link for the website is given in the description box. Now let's get started. Dental anomalies. Anomalies means abnormalities. And dental we know related to teeth, right? So dental anomalies are abnormalities that are related to our teeth. So dental anomalies are abnormalities of form, function, position of the teeth, bone and tissues of jaw and mouth. Alright, they are caused by multifactorial interactions. So there are a number of factors due to which the dental anomalies can be caused, and they can be genetic, epigenetic or environmental factors, and these anomalies are formed during the long process of dental development, that is when our teeth are getting formed, right? So the anomalies can be based on 4 types. First, anomaly of size, then there can be anomaly of shape, then there can be anomaly of number, and then there can be anomaly of structure. In this video I'm going to discuss these 2 types, that is anomaly that is based on the size and anomaly that is based on the shape. So first let's start with anomaly of size. In this there can be 2 types, that is microdontia and macrodontia. Microdontia means that the teeth are smaller than normal. As you can look at this word micro means small, right? So the teeth in microdontia condition are smaller than the normal sized teeth. As you can see here in these pictures, few of the teeth like this one, and in this all the teeth are smaller in size. Microdontia can be of further 2 types, that is generalized and localized microdontia. Generalized microdontia means all the teeth are smaller in size. It is a general idea that all the teeth are small, and the reason for that can be pituitary dwarfism. Whereas localized microdontia means only certain teeth are going to be smaller in size. If you look at this picture here, only this particular teeth is smaller in size, right? So this is example of localized microdontia, whereas in this one all the teeth are smaller in size than the normal one. So this is generalized microdontia. Example of localized microdontia is peg lateral teeth, which is basically our maxillary lateral incisor becomes cone shaped. If you look at this picture, this is the central incisor and this is the lateral incisor, and this is the upper jaw, right? So upper jaw is maxillary jaw, so the maxillary lateral incisor become cone shape. If you can see, this looks like a cone, right? So this is an example. This is called peg lateral and it is an example of localized microdontia. Then comes macrodontia, in which the term macro means big, right? So the teeth are larger than the normal size. It is also subdivided into 2, generalized and localized. In generalized, the reason for generalized can be pituitary gigantism. For microdontia it was pituitary dwarfism, whereas for generalized macrodontia it is pituitary gigantism. As you can see in this picture, all the teeth present are larger than the normal size. This is an example of the localized macrodontia in which only these two teeth are larger than the normal size, right? Next comes anomaly of shape, that is a shape of the person's teeth is changed, right? So first in this comes gemination. What happens in gemination is there is division of single tooth gum by invagination. So if you look at this particular area in this figure, this is gemination in which we have one root, right? But this portion, the white portion or the enamel portion of the teeth, we get to see a division in this portion of the teeth. As you can see, here is the division, right? So there is division of single tooth gum by invagination, and what we due to which what appears is like there are two teeth, right? One and two teeth. It looks like there are two teeth, but it is just a single teeth, right? So overall we can say that there is one teeth increased if we count this particular thing as two teeth, but the root is only one in gemination. Whereas in fusion, two teeth unite to form one teeth. If you look at this picture, we have two separate teeth and they have united with each other. So we have two roots in this case. We can say that there is a one missing tooth because two teeth have united and now it looks like just one teeth. So we can say that overall there is one missing tooth if we count this as just one teeth. There can be two types of fusion, complete fusion and incomplete fusion. In complete fusion, the roots are also fused, whereas in incomplete fusion, the roots are not fused, only the white portion is fused, right? As you can see here in this particular picture, this is an example of gemination. As you can see this invagination, right? And this is an example of fusion in which two teeth have, fused with each other. Then comes concrescence. If you look at this picture now, it is a condition of teeth with the cementum overlying the root of the at least two teeth joined together. So this root area, right, the cementum is present over the roots. Now if that cementum of the two roots fuses with each other, then that leads to concrescence. It usually involves only two teeth. The most commonly involved teeth are upper, second and third molars. So in this case the roots get fused due to fusion of the cementum. As you can see, this was tooth number 1, this was tooth number 2, and they have fused with each other. Then comes dilaceration. It refers to angulation or sharp bend or curve in the root or crown of a formed teeth. If you look at this particular teeth, you can see that the root has bended. There's a curve that you can see in this tooth, right? So this is known as dilaceration. This disturbance is more likely to affect the maxillary incisors. So this is seen to happen in maxillary incisors and occurs in permanent dentition. Then comes dens evaginatus and dens invaginatus. These are two terms. So in dens evaginatus, if you look at this particular picture, this is for dens evaginatus. This appears as an accessory cusp or globule of enamel on the occlusal surface between buccal and lingual cusp. If you look right here you can see that a globule of enamel is there, right? We can call it an accessory cusp, and this is present on the occlusal surface of the teeth between the buccal and the lingual cusp. So there are different cusps, right? So dens evaginatus occurs between the buccal and lingual cusp. The premolars are more likely to be affected than any other teeth. So mostly dens evaginatus is seen in the premolars. It is more common in men than in women, and it is more common in mandibular teeth, that is the lower teeth, than the maxillary teeth. If you look at this particular picture, here you can see this. These are the dens evaginatus that are seen in the premolars. You can see the extra cusp, right? Then comes dens invaginatus. It is also known as tooth within a tooth. If you look at this picture, there is a lingual pit extension that you can see. This is what happens in dens invaginatus. So there is infolding of enamel into the dentine. So it is a rare dental malformation that is found in a teeth where there is infolding of enamel into the dentine. The prevalence of this condition is 0.3 to 10%, affecting mostly the males. Then the teeth most affected are maxillary lateral incisors in 80% of the dens invaginatus. Maxillary lateral incisors are affected, followed by maxillary canines in 20% of the cases. Next comes talons cusp. Talons cusp is an example of dens invaginatus only. So that means there's going to be an extra cusp, but this time it is not in premolars. This happens in the incisors. So there's extra cusp in the lingual side. Lingual side means the side that your tongue is normally touching. That is the lingual side. Labial side means side that your lips are normally touching. Side of the tooth that your lips are normally touching. So extra cusp is found in the lingual side of the incisors, and talons cusp. You can see that in this picture here. These two are the talons cusp. It is also known as eagle's cusp or eagle's talon beak. Next comes taurodontism. It is a condition that is found in molar teeth of human, whereby the body of the tooth and the pulp chamber is enlarged vertically at the expense of the root. So if you look at this picture, this is a normal teeth. This is hypodontism. There can be three types of taurodontism: hypotaurodontism, mesotaurodontism, and hypertaurodontism. In normal condition, we have the normal pulp chamber. The normal volume of pulp chamber can be seen here. In hypodontism, there is slight increase in the pulp chamber. Here in mesotaurodontism, even more pulp chamber area can be seen, and in hypertaurodontism, even more pulp chamber can be seen. As a result, the floor of the pulp and the furcation of the tooth is moved apically down the root, as you can see. This is a radiograph for taurodontism. You can clearly see this one is having taurodontism. These are the normal tooth having normal amount of pulp chamber, but in this area you can easily see hypertaurodontism. Then comes enamel pearls. These are developmental anomaly of teeth, also known as anomalies, enamel droplets, enamel globules, enamel nodules, enamel knots, or enamel exostosis. So simply here you can see in this area there's a enamel pearl globule type thing that you can see. So this is enamel pearl. They are ectopic globules of enamel on the root surface. This is the root area, right? And here you can see this enamel pearl. Next comes shovel-shaped teeth. Lingual extension of lateral borders of incisors. This is seen in the lingual extension, lingual as I told, and on the lateral borders. This is a picture of a shovel-shaped teeth. This area is shovel-shaped, right? These are the lateral borders of these incisors. This is seen in mongoloids 80 to 85% cases, in caucasoids in 2 to 6% cases, and negroids 1 to 2% cases. Then comes cusp of carabelli. This is basically an extra cusp that is seen in the maxillary first molars. So the cusp of carabelli, or also known as carabelli's tubercle or tuberculum anomale of George Carabelli. It is a small additional cusp that is seen as the mesiopalatal line angle of the maxillary first molars. Here you can see this is marked over here that this is the cusp of carabelli. It is basically an extra cusp that is seen in the maxillary first molars. Then comes protostylid. A protostylid is a supernumerary or accessory cusp located on the mesial half of the buccal surface on the molars. Buccal surface is the surface that is touching your cheek area, right? So here you can see this particular teeth, this is a protostylid and it is seen in less than 1% population. So it is a very rare tooth anomaly. Part 2 video of dental anomalies. In my previous video, I have discussed about anomalies related to size and shape. In this one, I'm going to talk about anomaly of number and anomaly of structure. So you can go check out video for anomaly of size and shape. I've provided the link for that in the description box. If you want, you can go check that out. Also, if you want this PPT, then I've uploaded it on my website. You can go visit my website, download the PPTs from there. Link for the website is also given in the description box. Now let's get started. Anomaly of number can be of 3 types, that is anodontia, oligodontia and hyperdontia. Anodontia means that there is complete absence of teeth. There are no teeth present in that particular condition. If you look over here, you can find no teeth in this particular jaw, right? So this is anodontia condition. It is a genetic disorder. Then comes oligodontia. Oligodontia is a rare genetic condition which represents the congenital absence of more than 6 teeth in primary, permanent or both the dentition. It is usually a part of syndrome and seldom occurs as an isolated entity. So oligodontia means that there are more than 6 teeth are missing from the jaw of the person. If you look here in this picture, 6 or more than 6 teeth are missing, right? A teeth is missing from here, one is missing from here, here, right? So if 6 or more than 6 teeth are missing from the jaw, then that is a condition known as oligodontia. Then comes hyperdontia. Hyperdontia is a condition that causes too many teeth to grow in the mouth. If you look at this picture, there are so many teeth that you can see here in this particular mouth. So hyper means more, right? So hyperdontia means too many teeth. These extra teeth are sometimes called supernumerary teeth. They can grow anywhere in the curved areas where teeth attach to your jaw, as you can look here in this picture. And the example for hyperdontia is mesiodens and fourth molar, which are discussed in the next slide. First, let's talk about mesiodens. It is a supernumerary teeth that is present in the midline between the 2 central incisors. If you look at this picture, this is one central incisor, this is second central incisor, and you can see that here is a tooth present in between these two, and this is a mesioden. It usually results in oral problems such as malocclusion, food impaction, poor aesthetics and cyst formations. Then another example of hyperdontia is fourth molar. Fourth molar are supernumerary teeth located distal to the third, so they are present next to the third molar, and hence they are also known as distomolars because they are present distal to the molars, or also known as distodens. The prevalence of fourth molar in the study population was found to be only 0.32%, so this is very rare, and fourth molars occur with approximately equal frequency in male and female. So the chances of the gender having the fourth molar are approximately same, that is both male and female has equal chances or probability of getting a fourth molar. Fourth molar are more common in the maxilla, that is the upper jaw. If you look at this particular picture, here is the third molar, this is the third molar, and here is the fourth molar. All right, now let's talk about the anomaly of structure. So first comes dentinogenesis imperfecta. It is a disorder of tooth development. This condition causes the teeth to be discolored, often a blue-gray or yellowish brown color, and they become translucent. If you look at this and this picture, you can see that the teeth are slightly, transparent in this case, right? They are yellowish brown color. They are also weaker than the normal, making them prone to rapid wear, breakage and loss. Here you can see that the teeth are broken, right? Because why they are broken? Because they are weak, hence they are prone to breakage and loss, and they become tulip shape. If you look at this particular teeth, this looks like a tulip, right? So they are tulip shape. Then comes amelogenesis imperfecta. It is a disorder of tooth development. Then this condition causes teeth to be brown, opaque, discolored, pitted, grooved, and prone to rapid wear and tear and breakage. If you look at this particular picture, the tooth appear kind of brown, yellowish, right? Discolored, right? And they are pitted, grooved, and they are also prone to wear and tear and breakage. They are also weak. There are certain subdivisions of amelogenesis imperfecta. First, type 1 is hypoplastic, type 2 is hypomaturation, type 3 is hypocalcified, and type 4 is hypomaturation or hypoplasia or taurodontism. Now let's move on to the next anomaly of structure, that is Turner's teeth. It is a defect of enamel. Enamel defect in the permanent teeth caused by periapical inflammatory disease in the overlying primary tooth is referred to as Turner's tooth, also known as Turner's hypoplasia. So Turner's teeth is caused due to an inflammatory disease called periapical inflammatory disease, and in this the teeth, if you look at this picture, the enamel portion of the teeth is affected, and the tooth's thickness is decreasing, so due to which the sensitivity of the tooth is increasing. Then moving on to the next anomaly, that is Hutchinson's teeth. It is a sign of congenital syphilis. This Hutchinson teeth and the mulberry teeth both are a sign of congenital syphilis, which occurs when a pregnant mother transmits syphilis to her child in the uterus or at birth. So this will occur in a child when a mother is going to transmit the syphilis to the child during pregnancy. The condition is noticeable when a child's permanent teeth come in. The incisors and molars take a triangular or peg-like appearance. If you look in this picture, you can clearly see that the molars, incisors and the molars are peg-shaped or triangular shaped. Then talking about mulberry teeth, as I said, this is also associated with congenital syphilis, and it is characterized by multiple rounded rudimentary enamel cusp. You can look here very clearly that they are these teeth have started to look like mulberry fruit, right? And this happens in the permanent first molars. Here you can see they start to look like mulberry, hence the name is mulberry molar. Mulberry molars are physically defective permanent molars, and both these Hutchinson teeth and mulberry teeth are associated with congenital syphilis. Next, moving on to ghost teeth. Teeth in a region or quadrant of maxilla or mandible are affected to the extent that they exhibit short roots. So ghost teeth have short roots, they have wide open apical foramen, they have very large pulp chamber, then the poor mineralization qualities of the enamel is there and the dentine layers, and they have given rise to a faint radiolucent image, hence the term ghost teeth. If you look at this particular picture, the image that we get to see in the radiogram is very faint, hence the name is given as ghost. As you can clearly see that the root area present here, the roots were supposed to be there, but the roots are so tiny that we cannot even see them properly. So in ghost teeth, there are certain characteristics, that is they have very short root, they have very open, wide open apical foramen, they have very large pulp chamber, and there is poor mineralization of enamel and dentine. Then comes fluorosis. Dental fluorosis is a condition that causes changes in the appearance of tooth enamel. It may result when children are regularly exposed to fluoride during the teeth developing years, that is from 8 age or younger age. Now fluorosis can be very mild, it can be moderate, and it can be severe. You can see for the differences that will be there in the different stages of fluorosis. So I hope you understood all the types of anomalies, that is the anomaly of size, shape, number and structure. This was all about this video. Thank you for watching.